Prior to mitosis, the cell goes through the cell cycle, the longest of which is interphase. Within interphase, there is 4 processes: G1, S, G2, and the mitotic phase. G1 is when the cell prepares for cell synthesis, or the S phase. The cell replicates its DNA to have 2 exact chromosomes available during the S phase. Finally, in G2, it finishes growing and prepares for mitosis. The objective of mitosis is to produce 2 daughter cells from one parent cell with the same amount of chromosomes. This only can occur through 4 processes, the first of which is prophase, then metaphase, anaphase, and finally telophase. During prophase, the centrioles divide and begin to move to opposite poles, forming asters between. The chromosomes are uncoiled and the nuclear membrane dissolves. After prophase comes metaphase, in which the chromosomes double and attach to spindle fibers in the middle. During anaphase, the centromeres split, moving half the chromosomes to one side and the other chromosomes to the other pole. So there's half the chromosomes on one pole and the other half on the other pole. During this occurring, the cell membrane tightens, spreading the cells into half, spreading them. And finally, during telophase, the 2 halves split, creating 2 daughter cells with the same amount of chromosomes as the parent cell. Prophase, metaphase, anaphase, telophase. Meiosis has 2 stages, or processes of a sort, the first of which is prophase 1. In prophase 1, the centrioles move to opposite poles. The DNA forms homologous chromosomes, which pair together to form bivalents. The bivalents then duplicate themselves to make a tetrad. They cross over to produce variations of the genes. It then enters metaphase 1, where the tetrads meet in the middle of the spindle fibers. After metaphase 1 is anaphase 1, where the chromatids split half to each pole, so half of the chromatids go to one pole and the others to the other pole. During this, the cell membrane squeezes together, preparing the cell for telophase 1. During telophase 1, the cell splits and the nuclear envelope takes in the chromatids, thus ending the first stage. The second stage of the meiotic division onsets with prophase 2. The homologous chromosomes separate as the centrioles move to either opposite poles. Once this has been achieved, the cell enters metaphase 2, where the split chromatids line up in the middle of the spindle fiber on their centrioles. It then enters the stage of anaphase 2, where the chromatids reach their poles, and while doing so, the cell membrane is being constricted. Finally, in telophase 2, they separate to make 4 cells with half of the chromatids than its parent cell did. Prophase, metaphase, anaphase, telophase, prophase 2, metaphase 2, anaphase 2, and telophase 2.